So thanks again all for coming, be it speakers or the audience, to our first the second Klima webinar, because Rui sniped us with yesterday. But the more the better. So today is a networking event on popular demand in the Advanced Imaging Task Force. The point of today's event is so that people can present their research and capabilities and then connect with interested people to form research collaborations of any kind, be it shared research, shared applications for grants, brainstorming, academic exchanges, searching jobs, offering jobs. As long as you're networking, we are happy about it. We have 7 speakers scheduled for today. So the general framework of this is that everybody will present for up to 7 minutes their capabilities and research interests, everybody of the speakers. And then afterwards, we will disperse into breakout rooms where you can talk and interface with these speakers and each other. If there are any questions, please put them into the chat, and depending on if it's technical or organizational, me and the ESMRP office will handle this. And yeah, without further ado, I think we can start. So Rita is going first. And
okay, thank you very much. Let's share the screen. Can you see the screen?
Hopefully, yes.
Okay, so I'm representing our lab, Evolutionary Systems and Biomedical Engineering Lab. We are based at Instituto Superior Técnico, which is the engineering faculty of the University of Lisbon. So to give you an idea of where we are, yes, this is Lisbon, okay, and this is our campus. So it's a university campus. We actually sit in one of these towers. This is why we can see the other tower and nice view of Lisbon. In this group, we have 6 PIs. We actually have someone joining in October, but also Agostinho Rosa, the founding member, he's actually retired, so he's no longer quite as active. From this list of people, you probably, well, you know me and Patricia Figueiredo, who's currently actually leading the SMRMB. And so the 2 of us, we're very much interested in MRI, neuroimaging in particular, and myself also MRI methods acquisitions. And in terms of the Glimmer point of view, well, we have this collaboration ongoing with Hospital da Luz, with neuroradiologist Dr. Pedro Vilela, and we also have PhD José Maria Moreira, who's at the research hat of the same hospital, so it's a private hospital, and Teresa Nunes knows it quite well as well. And José's expertise is in machine learning, so he's been giving us quite a lot of contributions as well. In this project, where we're looking at the longitudinal follow-up of glioblastoma, so it's a clinical data set, we have our 3 PhD students here who are very much active in this interest group as well. So they are part of the communications task force, and Katarina and Marta. And so the idea is that, just to give you an idea how it compares with some public data sets. So we have retrospective data from glioma tumors, which some of the patients, so few, unfortunately, some exams go up to close to 5 years, but obviously we know that's unfortunately not the most often. But what we aim to do is, well, look at this data set. So if starting with the first tasks was, well, okay, we need to be able to segment the tumor somehow. And Katarina looked into, well, can we perhaps in some cases where we don't have the all 4 conventional anatomical images, if we could maybe use only FLAIR and T1 contrast enhanced, because these are the most informative. And so in this case, she used the BRATS 2021 data sets and tested 2 different architectures. And what we saw was that actually ResNet was better at segmenting the whole tumor and also the tumor core, but the SWIN performed better when it came to looking at enhancing tumors. So we proposed an ensemble model to combine these two results, and we could actually get quite nice results only with 2 images. Of course, if we have 4 images, that's great, but what we notice is in some of the data sets, this is not always the case. So this is preoperative, but of course, since we have a longitudinal data set, we want to be able to also do segmentation of the post-op. So that's a big challenge, because obviously we have all these other issues to deal with. So what we've been trying to do is, well, can we perhaps take advantage of models trained on the pre-op and do some kind of transfer learning to help us do also the segmentation on the post-op. This is something she's been looking at and also presented at this year's ISMRM. But obviously, we would like to continue and maybe think of some federated learning, some options that we can work on together. Other challenges, well, this is a clinical data set, so it has data acquired in many different scanners, not always the same manufacturer, different manufacturers. Protocol is not completely stable because there have been updates, upgrades of the software, etc. So having to deal with data harmonization, that's also a big concern, and this is something Marta has been working on. So at present, she's been looking at existing tools, namely combat and long combat, and trying to work out if we can get more comparable data that which can then be used to feed models to help us well, do all sorts of things we need to do. So the idea is to be able to predict what's going to happen to the patients, obviously. And so in that respect, Anna Matos has been busy looking at, in this case, she used longitudinal data from Lumiere, which is a public data set as well, and tried to compare different strategies for predicting if the disease is progressive or not. So according to the Rano criteria, and if you want to know more, you can check her preprint here. But there's still quite a lot of work to be done. Our results were still, this is just an initial attempt, so it can definitely, there's a lot of room for improvements. So what we want to do now, we also want to explore other data that we have. So we have diffusion data, perfusion, mostly DSC, but also some ASL data, and we have a prospective branch to this study where we want to acquire more ASL. Of course, Patricia is an expert in ASL, and we want to improve the diffusion and get some relaxometry data. Of course, if there's a lot of tasks here that we could collaborate on, so it would be great to hear your ideas and find out how we can work together. And from my side, that's it. So this is some of our group, and it'd be great to hear about your presentations as well and interact with some of the people attending. Okay, so I'll stop sharing now.
Thank you, Rita. Also very nicely in time. I appreciate it always. Yes, I'm tracking and stopping actually. So I will interrupt you if you take too long and give you a warning before. Okay. But you did well. So that's how I envisioned it. Yes. So thanks, Rita. And our next speaker is Ana Paula Candiota from the Universitat Autónoma de Barcelona. I hope I didn't butcher any pronunciation too much. Also for the next speakers, I'll try. And if you're ready, you can go. But I can't hear you right now. Are you muted, maybe? You need to unmute yourself.
Yes, but I am not
Yes.
in my screen. Sorry.
We see your full screen.
You see?
Yes.
Oh, so that's perfect. I'm sorry about this technical problem. That's why we did the technical tests before. Now I'm seeing in the other screen. And then my talk still has a title, which is Assessing Response to temozolomide therapy and TMA, tumor microenvironment changes in preclinical brain tumor models. So it means that in Glimmer, we are in the preclinical side of the question. I did not really prepare some things about the group as Rita. I was not expecting to do this, but I'm the only PI, and we are a few people for the moment and willing to grow. Historically, the group background was to apply NMR-based approaches, especially spectroscopy in tumor. At that time was mostly clinical brain tumor diagnosis, grading, and now for therapy response assessment. But currently, this part of the group is focusing in preclinical immunocompetent brain tumor models. And we test new therapeutic approaches, we assess the response to therapy, and we were trying to validate what we are seeing. In this case, this research branch appeared more or less with Rui, so it's due to Rui or Rui's fault, depending on what do you want to assess. I will not repeat the whole history because I think either Glimmer and the ISM-RMB people have already seen, but it's based on MRSI, which is spectroscopic imaging acquisitions in control and temozolomide-treated mice. In principle, most of the results are based in GL261 immunocompetent brain tumor model. So as far as almost 10 years ago, so we saw differences between control and treated responding mice even before. So these differences appeared before tumor volume changes. So they were telling us something about the different metabolism of what was going inside. And then going a step beyond, we moved to multi-slice acquisition to have this heterogeneous information about the tumor. And this presented in a longitudinal study, this presented like it was not static. Even if the volume was static, this was changing in a cyclical way, more or less every 6 days. And then an initial validation showed us when the treated responding mice had increased lymphocyte infiltration and increased macrophage infiltration. So if you join both things, this frequency of changing this pattern, which is shown in color because it's automated classifier transformation in colors, it transforms patterns in colors. So when you see red, it's control of non-responding, and you see green is responding. So if you join both things, so we imagine that it was somewhat related to the cancer immune cycle, which has a 6-day cycle, and it could more or less correspond to these waves of immune cells trying to attack the tumor. Then if it was important, what if we adapt our cycles and just do the temozolomide trying to respect these cycles? At this point, we still see this every 6 times oscillation, so it was confirmed, and we ended up with cured mice. So it was not happening before. Cured mice. Not only that, mice were cured, and we were not able to generate tumors in those mice again. So they got some kind of immune memory, which is not fully characterized, but it happens. Then more extensive validation was done. It's not everything on those slides, but these are in the references, is macrophages, carbonic anhydrase changes, metalloproteases, PD-L1, everything related to this tumor microenvironment changing. And we associated it with this responding pattern. So we try to go steps beyond and characterize. The criticism that we usually receive is that the GL261 is very immunogenic as itself. So we were just navigating in a very favorable scenario. So we are putting on 2 additional immunocompetent models, which are CT2A and SB28. They are quite different in both mutational load, PD-L1 presence, MHC expression, the overall immunogenicity. So it's all is going to be characterized, is in process. So it's not published, that's why it's not here. But as you can see, the differences in response, even with the most favorable schedule that we have there, these responses are very different. This is in preparation as well. And we tried to see first if our system developed to classify gel-261 worked. It's more or less worked to DCT2A, so it's able to catch the transient response. It can catch DAS-B28 during poor response, so more or less this classification system, which was developed for gel-261, seems also work for the other models, but somewhat loses when we are trying to characterize untreated tumors. So it has to be retrained.
You have 1 minute left.
Yes, thank you. I'm chronometering. And now the work which is in progress, which is not here because it's not published yet, is the whole characterization of the 3 cell lines regarding immunogenicity, release of immunogenic signals, immunohistochemistry of different factors of stemness, and pathways of resistance. And we are several things that I think we can intertwine and interact with other Grimmer partners, and we can just talk about it in the post-session discussion. That's all from my side. And thank you, the funders, very important.
Thank you very much. That's very interesting. If I have time for some MRSI questions later, I'll pop into your room.
Stop sharing.
And our next speaker is someone new to the Glimmersphere, Daniel Cocking from the University of Nottingham.
Thank you.
If you're ready to go, that's not your full screen.
There we go.
That's your full screen. Awesome.
Brilliant.
Yeah.
Thank you very much.
When you're ready, go on.
Brilliant. Thank you very much for the introduction. Yeah, so I'm a postdoctorate at the University of Nottingham, working in a very multidisciplinary team with Dorothee, Auer, who will be hopefully presenting some slides later on and more the biomedical applications, but I have more of a physics background, so I'll just be presenting some of the work that we've done and that we're planning to do in terms of the physics. So a lot of the work we do involves deuterium metabolic imaging, but overall we aim to work with spectroscopy on gliomas and glioma subtypes. So why do we use deuterium? Well, it has a low natural abundance, which makes it ideal for tracer studies. It's got an inherently low SNR and short relaxation times, which allows for lots of averaging to recover lots of that SNR. And then we've previously presented work at RSMRM showing that we can use CSI measurements to acquire data across the whole brain. And the results that we get appear similar in concept to positron emission tomography. We've done this in healthy groups now, and we've just started doing patients with mainly glioblastomas, but we plan to extend to quite a large tumor cohort. So far, we have scanned 6 patients, 3 of which had glioblastomas, 1 primary CNS lymphoma, and 1 oligodendroglioma, with voxels identified within the tumor region compared to the rest of the brain, from sort of a very coarse mask. We've recently scanned a new patient that had a IDH wild-type mutated glioblastoma. And this is sort of the data that we end up getting. So we get quite a coarse grid across the whole brain. Each voxel contains a spectra which is fit using a MATLAB toolbox that we've adapted from working with phosphorus and carbon. Quantitative analysis on this data is yet to be applied, so looking at identifying that tumor region. As of this morning, we actually got that through, but it's yet to be added to the slides. But we're very excited for the results specifically from this patient, especially in their sort of lactate GLX signals. We have been giving our patients 7 labeled deuterated glucose, which might be different to the usual deuterated glucose you're usually used to seeing. This gives us inherently more SNR across each of the metabolites. It's quite a bit more expensive, but we're very happy with the results we're getting from the increased water signal to potential increases in lactate and GLX. So what are we doing to improve our sort of data acquisition? So far, all of our data has been acquired using a single channel birdcage coil that we purchased from Rapid Biomedical. And all of our scanning so far has been performed at 70 on a Philips Achiever scanner. But recently we've purchased a 8-channel proton, 4-channel deuterium channel coil array from Coiltech. And we're just about getting over the last few niggles of setting that up and getting all the coil combinations correct. And then also you might have heard that we've got building work starting soon on 11.7T MR scanner that's going to be built with a Philips backend, and it will be deuterium capable, and we'll have a proton deuterium RF coil purchased with it, as we have an agreement set up with Coiltech. So on the scanning side, what are we aiming to improve? So obviously we're using a repeated CSI measurements with Hanning weighted acquisitions, but we're hoping to start developing some faster MRSI acquisitions that have been seen to be more data efficient for proton and deuterium signals, such as FID MRSI and concentric ring trajectory MRSI, which we're very excited about. And then we're hoping that using these and other spectroscopy techniques, we can implement quantitative exchange labeled turnover, where our deuterium enhancement in sort of tumor regions can be indirectly detected by changes in the proton detection. And we're hoping to be implementing that soon with semi-laser sequence with Goya pulses. We've recently had a digital upgrade on our 7T, so we're excited to see the benefits we'd get from enhanced B1. But yeah, thank you for listening. And Dorothee will be presenting some of the work on the more biomedical applications. But yeah, hopefully we can engage in some much needed conversations around this sort of topic. Thanks.
Thank you. I see Dorothee has joined us. Dorothee, for your information, you are the third speaker coming up. So you still have some time to get into. Because our next speaker is Yaara Erez from the Faculty of Engineering from the Bar-Ilan University.
Hi, everyone. Thanks for the opportunity to present our research and interest in collaborations. I'm based at Bar-Ilan University in Israel. This is our Faculty of Engineering, although my background is computer science, psychology, and neuroscience. This is what we work on in the lab. Glioma imaging is only part of our research. We specialize in signal processing, all sorts of signals, intracranial EEG, fMRI, EEG, other physiological measures. And we look at cognitive processes, specifically attention and executive function and the related brain networks. We also work on real-world application in the industry and the clinical ones, the clinical domain, and this is where our work with patients with glioma fits in, although it takes up quite a large proportion of our work. Our interest in glioma imaging started when I was still based at the University of Cambridge. And we initially started with functional mapping during surgeries for patients for the surgery for tumor removal. trying to see whether we can use intracranial EEG to improve clinical decision-making as to which tissue to remove or to keep. So we did quite a lot of work along these lines during surgeries, and we keep doing it now here in a collaboration with the Soraski Medical Center. For example, one of the findings is that we were able to show that we can distinguish between executive control regions and adjacent regions using ECOG, and we are now using this in real time as part of the clinical procedure at the hospital. These are awake surgeries. We also developed some tools to assist with pre-surgery planning. This is a prototype that we developed that incorporates pre-surgery MRI, fMRI, and DTI on a 3D printed brain. And more generally, we use multimodality data sets both before and after surgery. So we have longitudinal data. We use MRI, fMRI for both rest and task, DTI, cognitive assessment, and intraoperative ECOG or intracranial EEG. And what we are aiming to do is to support the clinical decision-making process, but also to predict cognitive outcome, to understand disease progression and how it affects brain networks. One example is work that we did that combined ECOG with REST fMRI in one of our cohorts, and what we looked at is whole brain connectivity with fMRI and linked connectivity with a dorsal attention network and a tumor area to post-surgery cognitive outcome related to attention. We also look at several other things. One other example is connectivity with here again with REST fMRI before and after surgery. One of the things that we found is that there is a laterality effect for connectivity, specifically for the FPN. This incorporates again ECOG data and fMRI. So most of the electrodes, the ECOG electrodes, were placed in the FPN. We don't yet know whether this asymmetry is specific to the FPN or just as a result of the location of the electrodes. But these are the kind of things that we look at. For collaborations, for anyone who is interested, we are interested in pre-surgery planning, predicting cognitive outcome, intraoperative mapping, which is not written here, but it's much of the work that we do, although that doesn't involve the MRI, but electrophysiology. We are now getting into radiomics, and we are very much interested in longitudinal measurements where we can follow up the brain processes. Thank you for your attention to the patients, and this is us and funders.
Thank you very much. Lots of interesting points in there, actually.
Happy to talk.
And you also were very nicely in time. So the next one is Rui Simões from the I3S Porto.
Can you hear me?
Yes, but that's not your full screen, or it's not your full screen yet.
Not yet.
But we can hear you.
How about
yes.
now? Okay.
Great.
Cool. So, hello, good afternoon, everyone. I'm Rui Simões, as Gilbert introduced, and I'm based in Porto, in Portugal. So Rita is here in Lisbon. We're a bit norther than that. So specifically at the university campus of the University of Porto. So I3S is located here, and it's an umbrella of 3 different institutes that cover different types of research. So the preclinical MRI is located here at I3S. And the interesting feature of this environment is how close it is to the reference hospital in the north of Portugal, which is Hospital São João, and also the close proximity to the Portuguese Institute of Oncology, and the fact that there is a consortium called the Porto Comprehensive Cancer Center, which is one of the few in the world and in Europe, of course. So this gives us a certain leverage for the type of research that we do here. So the preclinical MRI at F3S is relatively recent. I moved here in 2022, and in 2023 we could start working, but it wasn't, and we installed a 3 Tesla Bruker scanner. So it has a clinical magnetic field and therefore promotes more translational MRI research, if you will. We have high power gradients, which are convenient for different applications. And since last year, we had a functional cryogenic coil that boosts our sensitivity and with which we can work with a sensitivity around 9.47, give or take. So, which is crucial for mouse neuroimaging, as I will show you. So what we are promoting here in the context of glioma research is the neuro-oncology hub, giving its first steps. So by now, and before I arrived, it was already pretty well established. The patient-derived organoids being generated from brain tumor biopsies, and this pretty much in the context of pediatric brain tumors, and kickstarted by Jorge Lima and led by Joana Peixoto. So in the context of different projects, national and international, more recently, now with adult tumors. And the idea here with the organoids is that is to complement what is currently done. So for brain tumors, of course, the biopsy, there's a biopsy resection, and there is a pathological, a diagnosis based on the histology, which will help in the clinical decision making. And what this brings is complementing this branch by doing organoids, so they are dissociated from the main sample, cultured in multi-well plates, which then enables an extensive molecular characterization and even drug testing, which can have high throughput, and the whole point is facilitating precision medicine. So what we are doing from here and starting now, as I said, is generating mouse patient-derived xenograft models of these tumors and follow them up longitudinally with MRI to assess treatment efficacy and progression, invasion, and so on, with the idea of establishing markers that can be translated to the clinic, facilitated by the same magnetic field strength. So the first results that we have, and I showed this recently, this month, they're quite recent. So it's the longitudinal progression of a human cell line. In this case, it's a commercial cell line. It's the first results. We have PDX growing, no results yet. As I said, it's recent, but you can see very well here the tumor growth at different weeks post injection in this case. So growth is very obvious. When you do spectroscopy, in this case single voxel, it's very obvious that here there starts to be some infiltration, so you start to have some lactate in an otherwise pretty normal spectral pattern. And inside the tumor, of course, you have the typical features of aggressive proliferation with high choline compounds, a neuronal loss, low NAA, and glycolysis with a low glutamate and high lactate. Very interesting too is to see the good performance that we have for microstructural images. In this case, with diffusion kurtosis imaging fitting of DTI acquisitions. So at 1 week after injection, we see well the injection site and the loss of the natural microstructure of the brain, rupture here in the corpus callosum. So then it progresses a week later, and at the time when we sacked the animal, we already had a reorganization here of the microenvironment, very peculiar, also seen in the fractional anisotropy maps, which show more anisotropy here inside the tumor. So we're following this up as well, focusing on the tumor images with the fitting of the DTI data, in this case with the Sandy model. which will help us identify cell shapes and sizes. And well, we also have a problem, which is low SNR here at the bottom, and this is being tackled. We have better results now, better pipeline here dealt with João Zamit. So I'll be showing those results soon. But so far, we are seeing already what we expected from this cell line. So the cells are more elongated, as you can see from the estimations of stick fractions. So the normal cells are more spherical, as you see here in the sphere fraction. I'm being a bit chaotic, sorry. But in the cortex, you have more spherical cells.
1 minute left for you.
They're more elongated. Okay. So they're also big, and you can start seeing invasion, at least we think we are. And this is apparent also in different slices. So we are investigating this and validating it with human PDX models, as I said, and with more sequences that are being optimized and to assess treatment response. So there are different opportunities for collaboration from the clinical side, preclinical and computational, that we can discuss later, like the biological meaning behind the MRI sequences in the clinic and best protocols to assess invasion, also informed by histology, and the translational potential for more advanced protocols at ultra-high fields, and from the computational sides, from data processing to predictive models of DME evolution. Sorry, and that's it. Thank you.
Perfectly on time. Thank you, Rui. And our next speaker is, our next speaker is Dorothee Auer from the University of Nottingham. Yeah, I can see the screen nicely, but you're still muted. We can't hear you
Okay,
yet.
sorry. So
Now we can hear you.
Good afternoon. So I would like to give sort of some addition to the description of what's going on in Nottingham from Daniel. And while we are focusing on the metabolic imaging as a wider team, and I'm very happy to make introductions to the wider clinical team, wider MR physics team, biomedical and oncology team. Why can't I now get... Okay, sorry for that. I don't know which button it actually worked. So the only thing I would like to say is of opportunities that we have. We have a large Nottingham-based NHR-funded biomedical research center, which obviously doesn't have a cancer theme, but we do cancer in the MRI theme, and we have opportunities and scanner opportunities. As Daniel said, 11.7 Tesla is coming. We have several research clusters and also an intraoperative scanner, which is of interest. The objectives are probably what most people in this call have interest in: biology and mechanism of disease subtypes, and we want to have better tests so that we can help to identify innovative treatments. And from the four main areas is basically the brain tumor microenvironment and the prediction of tumor resistance and recurrence, where we see the most advantage in the metabolic imaging. I'm not going to rehearse that because you're probably all familiar with that, other than to say it's 100 years of Marburg effects discovery, at least 100 years that it was translated in English, and it needs to be refreshed. So there are some clearly cell-wise typical features, but this is not a full feature. And what we are looking at with our various imaging techniques and spectroscopic techniques is always part of the picture. And I've just highlighted here so we don't forget that the subcells, when we look at the worst of the dismal prognosis of the glioblastoma, there are 4 subtypes that are transcriptomically described, but 2 of them have actually a particular feature space that is described by Grafano recently in terms of metabolic alterations. And from these, there's a very small and tiny element that is MR detectable, whether you look at the pools of the spectroscopy or at the enrichment with the deuterium or other labels like DNP labels. So there is, it's always, we shouldn't forget, there's a big challenge that we have in front of us. So 3 topics I think many of us on the call are interested are these metabolic phenotypes at baseline predictive of treatment? And is the metabolic response to treatment indicative of the future clinical response? And ultimately, if you have the right metabolic subtype, can we then highlight a particular novel metabolic target that can be stratified? You've seen, and just very briefly going over what we've observed so far from Daniel, that we have a couple of first cases. Surprise so far is maybe there's one mitochondrial subtype where we have a much enhanced TCA activity. Most other cases, actually, regardless of the histology, have a downregulation of TCA, which is part of Warburg effect, but not much in the way of the glycolysis. And that's probably because it's far more challenging than looking at cells. It's composite at macroscopic scale. It's different cells, different genetics, different transcriptomics. There's a heterogeneity of everything we know about the microenvironment, but there's also the macroenvironment. And we really don't know what we are looking at the moment. So we need to be aware of it's only a macroenvironment readout that we have. And we are always specific to particular pathways. So our general findings of lower lactate enrichment might just be requiring a new model for us to understand. From the referencing we've done, we've partially but not fully addressed the individual variability. We need to do more to become more robust. And we do see some individual results already with reduction of TCA where it makes sense, but also like the PCNS or the mitochondrial subtype. This is the case that we find really exciting. As Dan has said, it's probably radionecrosis, where we probably see very low GLC uptake here in this area. We see reduced TCA. We also see lower lactate. So again, this partial Warburg, not a full Warburg. And really interesting, because that's what PET sees. HGO is almost normal. So what are the metabolites that we don't detect? We detect it overall with HGO, with the metabolic water pool that's enriched, but we don't detect it with the other pathways. Is it the pentose pathway? Are there other pathways? So we have more questions and answers. And to summarize, if you have more questions and answers, we need collaborators. We need other people who help us. There's a lot that we need to do better in the methods optimization, as Daniel has said, but we are now at the moment just trying to get larger groups here so that we understand these discrepancies that we observed and hopefully derive metabolic signatures, very similar to what I've heard that that other groups are doing, but we try to do it all in humans rather than outside of sort of in vivo models. And I think ultimately, if we've understood it better, we can then hopefully share protocols, develop novel probes, and also multi-site protocols for future human clinical trials. And all of that requires a lot of money. So that's why I think a European joint grant application would be fantastic. And I'll leave it here. Thank you very much for listening.
Thank you. And that leaves me to round out the speaking.
I will check your time.
I'll check my time myself, but you can also do it. Let's double check me. I need to adhere to my own rules. Okay. Do you see my screen? And my screen works. Okay. And I'm also ready to go. So I am from the Medical University of Vienna, just currently sitting in Oxford for a few more weeks, but I will return in a month to Austria. And I will now talk about our application of spectroscopic imaging to gliomas and a few other things. Vienna is a site of 7 Tesla MRI since 2008. We have a very old and very big and bigly shielded magnet, which we hope that in the future we can replace with a more modern 7 Tesla one that should also improve clinical research a bit. But we have shown that we can get a lot of funding and publications out of these MR devices. MRSI itself combines techniques from imaging and spectroscopy to give us voxel-resolved insights into specific molecules that are well traceable. That already differentiates gray and white matter. It also differentiates lots of pathology and tumor types and could lead into insights into tumor microenvironments. Example molecules could be glutamate and glutamine. They also show very nicely why we need high fields for this, because as you can see, these spectra are pretty similar. And if you have low quality data or too low fields, it's very tricky to separate some of these molecules, and especially glutamate and glutamine are of interest. We know glutamate has roles in the infiltration of gliomas and glutamine in its metabolism. Clinically available MRSI, so on the vendor side, is sadly very undeveloped. In the case of Siemens, they are essentially selling the same stuff since 25 years, so you cannot expect high performance out of this. So when you're using this, you might be able to get a few voxels in your tumor if it's in the right place in the brain. And essentially, you look on things like, is there less NAA and more choline, and do you see lactate? And that is pretty limited to resolve tumor microenvironments. In Vienna, the big team, have developed a concentric ring trajectory-based sequence with FID acquisition that is very fast. So we're talking about 80 times faster than phase encoding that allows high-resolution 3D mapping of metabolites. And this we have also applied to gliomas. We have a few publications out on this. My publication on peritumoral glutamate and glutamine should come out over the next few weeks, got accepted last week. And we can see metabolic heterogeneities in tumors, spatially and metabolite-wise. And at lower fields, one has issues to map glycine and myoinositol separately, or glutamate and glutamine. And if we separate them, we start seeing differences happening. We have not yet fully understood them, but we are working on that. We have, for example, also seen that glutamine and glycine maps correspond better to PET, to amino acid PET than the standard choline, which is not that surprising considering that we are at least closer to the metabolic processes mapped in PET. And now I want to round this out with a few research foci that we have. So a big part of my group looks into the 7 Tesla-mers of brain tumor data. Applications we have explored so far are, apart from spatial resolution, also aspects like, can we predict certain markers like IDH local over the whole tumor and patient, or locally moving towards tumor microenvironments. Recently, we also had some nice data on predicting 5-ALA fluorescence neurosurgeons use. And as I mentioned already, looking at what happens in the peritumor, because we actually have the spatial resolution and coverage to do this in a way that you can't do with single voxel spectroscopy. And I have several PhD students working on this. Also, to improve the methods, we are working on quantitative MRSI. So we are trying to go from ratios to concentration estimates, even in a tumor. For the non-experts in MRSI, that involves a lot. So quantification in concentrations involves lots of assumptions and additional data, and is tricky because most of these assumptions are made for gray and white matter and don't really work that well in tumors. But we are approaching this and have some preliminary data that we showed at ISMRM on this. And what you can quickly see on this image, there is a difference between ratio maps and concentration estimate maps. And that might matter when we suddenly see a hotspot appearing next to the tumor that we don't see on a ratio map. But if that means anything clinically usable, is still open. We also have an interoperative MRI where we are working to introduce spectroscopy as well, but most interestingly, interoperative resting state of MRI. So we can also look at the points of brain networking directly before and after resection, be the tumor or epilepsy. So we can get some very nice data on this, and we now have developed some methodology and validated it in some easier cases like colostomies. And our institute, not me personally, also does the metabolic imaging, and we have shown a lot of very nice method development to boost speed and resolutions, either, as Daniel already mentioned, with direct or indirect measurements of this. So if you are interested in the TRM, you can also come to my breakout room on this. And my personal vision is to combine techniques like 7 Tesla and interoperative MRI on the long run to enhance neurosurgery, to maybe give neurosurgeons a TME plus infiltration percentage map, maybe with some added functional information and information from other modalities to improve planning and therefore surgical outcomes. And especially with maybe better therapies in the future, be the targeted molecules, better radiation or something, to really significantly improve glioma outcomes. And final points, what can we do? We have 7 Tesla MRIs. We also have 3 Tesla MRIs. We have an interoperative free Tesla MRI we can run research on. We are specialized on MRSI and deuterium, not only, but these are some of our main fields. And we are integrated into a large hospital, so we are connected to neurosurgery, neuroradiology. I should have copyrighted that. Neuro-oncology. We also have a PGI research center, so we have opportunities for many combined studies that are necessary nowadays. Of course, this requires a big team, so I acknowledge all of my colleagues, and with this, I'm ending on time perfectly.